This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1095, recorded on March 8th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. Well, today is a beautiful, beautiful spring day. There's only one thing wrong with that statement.
It's not spring.
March 8th. It's nowhere near spring. Well, it's supposed to be spring, but this is very, very early. We have a tree that's leafing out across the street from me that I can see. It's already green. We just hope that the Polar Express doesn't hit us again, which it could.
It could.
Good.
Here in New York, it's 12C and sunny.
Yeah.
Also joining us from Montreal, Canada, Angela Mingarelli.
Hello, everyone. It's actually pretty nice here, too. It is, I wrote it down, 44F and 7C. And it's sunny. I closed my blinds because if not, there was going to be a lot of glare. But I went for I went for a walk with my dog this morning. It was beautiful. And it's kind of scary that March 8th in Montreal, there's no snow. There's only like these little patches of dirty, like gross snow. But we're supposed to be getting, I think next week, by the end of next week, a snowstorm. I know Newfoundland was calling for 50 centimeters. So East Coast Canada. And we're supposed to get like tail ends of that, but I think spring is more or less here, which is also kind of scary because normally we'd have a month left of winter.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 54 Fahrenheit, beautiful, as Dixon said, spring day, lovely day for April, and here it is.
And from Austin, Texas, Rich Condit.
Hi, everybody. I think I'm in a similar advanced spring condition here. We have 84 degrees, beautiful day. We had a thunderstorm roll through this morning, but then it cleared up after that. I will say that tomorrow's high is supposed to be 63 degrees, 20 degrees cooler than today. So we're swinging back and forth, but I will also say that the bluebonnets are up. That's a big deal in Texas, okay? They are the state flower, and a wildflower that is just all over the place, and the harbinger of many more other sorts of wildflowers to come. So this is a great time of year. I don't have quite enough experience to be sure of this, but it does seem early. I think of bluebonnets as April.
Well, I'll be heading your way tomorrow. I'm on my way to Austin, Texas.
Yeah, aren't you guys doing a TWiV together?
We're doing several things. We're doing a TWiV 100 on Sunday at the Science Mill in Johnson City. And then we're doing a TWiV Monday, 11 a.m. at the University of Texas with Jason McClellan. And then I'm going to be doing a session at South by Southwest on Tuesday.
Cool.
So that should be fun. We're
going to have fun. There? The drive to Johnson City is going to be fun. You get to see lots of bluebonnets and stuff like road trip, man. We've done this before.
Yeah.
Vincent, you have to get some barbecue and send it to us.
Yeah, right. Barbecue. So I'm looking forward to that. That'll be fun. It's spring break next week, so I don't miss any. Well, that's why I'm going, because it's spring break and I don't have classes.
Should be going to Padre Island.
That's not for me. I'm a little too old for that. Well, actually, I'm not too old for anything, but I don't want to go there now.
I think almost always since having been to Austin, because it's spring break, I've been absent for South by Southwest. So I will get to see it in full bloom this year.
Yeah.
It's a madhouse, I think, but we'll find out.
That's what I heard. A couple of announcements for you. We have 2 meetings that tell you about. The first one is all about classifying viral subspecies. COVID-19 pandemic highlighted the importance of collaboration in tracing and classifying viral subspecies for public health responses in preparation for future outbreaks. BVBRC, CDC, NCBI, and NIAID, all these acronyms, will host a collaborative workshop on viral subspecies classification. It will bring together leading experts to review existing classification schemes, develop best practices, and equip researchers and public health professionals with knowledge and tools needed to address future outbreaks and potential pandemics. The workshop is April 8th through the 10th this year, 2024. We'll put a link for registration in the show notes. You can register and go to that. And then we have another meeting in Australia. I've been talking about viruses of microbes, which is happening, I think, in June, right? And there's another one in September, Options 12 for the control of influenza. It's like the Super Bowl. They have Roman numerals for this meeting, except we're at 12. September 29th to October 2nd in Brisbane. We'll put a link to that as well. We're going to be doing a TWiV. Yeah, so I'm going to Australia twice, 2 separate times. I'm not staying there for a couple of months.
So you're making 2 round trips to Australia. Okay.
I am. I know. My carbon footprint is off the scale.
Yes, it's
You're
pretty
going to get
massive.
2 springs also.
Your circadian rhythm's going to be all out of whack.
Yeah. Yeah. So we're going to do a TWiV immune because I'm going to be there. Kathy Spindler will be there, and so will Steph Langle. And this is supported by the University of Queensland. So we're going to go do that. Now, why should you be interested in the meeting? It has a focus on underserved populations in respiratory virus research. You're going to hear a discussion with a Nobel Prize-winning scientist. You can have networking events for early career researchers. There's going to be TWiV/immune. And the most important thing, you can cuddle with cute Australian marsupials right at the meeting site. Huh. What do you think of that, Angela?
Sold, definitely. If there's koalas, although koalas have certain strains of chlamydia that you don't necessarily want to
they
get.
have claws too, right?
Sharp claws.
You can clap at the koalas, yes.
Just don't pet the platypus. They'll pet your back with their poison glands.
What else is a marsupial? Is a kangaroo a marsupial?
Yes, kangaroos, opossums, everything
Maybe
in
they have
Australia
some of
is a marsupial.
those tiny kangaroos, right?
Well, everything in Australia is
wallabies.
All the mammals in Australia that weren't introduced.
That's right. That's right.
Accidentally.
And some of the people of Australia are so.
Wallabies are adorable. Please take pictures with them, Vincent.
They are the wallabies.
Okay, we'll do. All right. So also the day before the meeting at the University of Queensland, that's Saturday, September 28th, they're going to have a school for influenza, a 1-day prep course to get
I think our local elementary school does that.
fluent in respiratory virus research. And early bird registration ends June 13th. So go to the website if you want to go to this meeting. And don't forget to buy a copy of Dixon's book, The New City. Learn all about what Dixon thinks the new city should look like.
And by the way, Dixon, my daughter, so Sophie stuck her head in the office and said, "You know Dixon Despommier, don't you?" Yes, he's one of the co-hosts on Twitch. She said, "I'm just watching a video about vertical farms, and they're talking about that." And apparently this is for her AP Environmental Science class. She's
so cool.
She's learning about vertical farms.
Tell her to call me. I'll talk with her.
So that's the thing.
I told her to read your book.
Agriculture has a broader reach than viruses, right, Dixon?
Oh, I'm afraid so. But
that's okay.
A lot of viruses affect agriculture also, though.
And viruses affect non-humans. Agriculture does too. Yeah, I think it's a tie.
All right, it's time for In the News, courtesy of Amy Rosenfeld. Our first article, heterosexual transmission of smallpox. So this is emerging infectious diseases, clade 1 associated Mpox cases associated with sexual contact, Democratic Republic of the Congo. They're reporting a cluster of clade 1 Mpox virus infections linked to sexual contact. Case investigations were done, PCR confirmed infections, and this genome sequencing indicates they belong to the same transmission chain. And so the point here is that you're thinking, wow, I thought it was sexual contact already, right? Well, this is now a different clade because the one that caused the outbreak was clade 2B. So now this is, and there are two clades, clade 1 and clade 2. And clade 2 is further subdivided into two subclades, 2A and 2B. And 2B was responsible for the 2022 epidemic. So clade 1 can be spread sexually as well. Any comments on this, Dr. Condit?
That does it. You got the bottom line there.
All
Beyond that, it looks kind of like in a way, the sexual transmission of the clade 2 viruses. Okay, so.
That's one of our papers today, in fact. We'll talk about that. We have an epidemiological update from PAHO, the Pan American Health Organization. Oropouche in the region of the Americas. That's oropouche virus. We had already talked about this previously, and now we have 2,104 positive samples in Brazil, and now in Peru, 146 cases. So the previous time we reported this, it was Brazil. Now it's in Peru. So it's moving.
Probably in other places if it's in those two places.
Oh, sure. Oh, sure. Okay. This one is for Angela. This is in PLOS 1. Oterid gamma herpes virus 1 in South American fur seals and a novel related herpes virus in free-ranging South America sea lions. Prevalence of and effects of age, sex, and sample type.
Yeah, so this was just them discovering. Yeah, it was just like the discovery of a new gamma herpes virus, I believe, like from what I read, in this population of seals, which is cool, and sea lions. Well, I mean, cool. Not really, because it can cause carcinomas on these poor animals, but urogenital carcinoma specifically, I think. Which I've actually seen some of these firsthand doing necropsies on, not in seals per se, but other herpes viruses, gamma herpes viruses in other cetaceans. In pilot whales, I've actually seen them. When I was in vet school, we did necropsies on beached whales that had these urogenital carcinomas as well. Pretty frequent in marine mammals.
Since seem to know about sea life, Angela, why do humpback whales smack the water with their fins?
Why? I don't know if there's anything. I'm sure somebody, cetacean specialist, would know this. A whale scientist specifically.
The humpbacks, they breach and it looks really cool, right? But then they also come up and they stick their one fin up and they smack it on the water. I've seen some videos of it recently. I bet it's to get rid of parasites, ectoparasites, right?
Or it's a signal.
Or it's just some sort of, because I know that the small whales, they'll start, like the calves, they'll start breaching excessively just to gain like strength in their muscles to like they're
working out,
smaller and they're younger. Literally, they're working out. But when they're older, a lot of it is also like when they're happy. This is what is inferred. What we think is that like when they're happy or like they start breaching, and just like with dolphins.
If you're happy and you know it, clap your fin. Yeah.
Exactly. But specifically like the dorsal fin slap, I'm not sure if that's anything specific. If anyone knows, please email us. I doubt I could Google that answer quickly anyway. So.
I think it makes sense if they're happy. Like you can imagine that. But addiction, you don't think it's to get rid. Sorry, go ahead.
No, I was just going to say that they're extremely intelligent. Like these are sentient beings that have, like when we compare brain mass to body size, they're almost, some of them smarter than us. So they're extremely intelligent. And they communicate in ways we don't even understand apart from echolocation. So this could just be like some sort of demonstration of happiness. Ectoparasites.
Would you get enough shearing force on just slapping a fin down?
No, I think the parasites, at least that I know that they have, are so small that I don't think that would make a difference. There's not like large, like some fish will have like very large, like acarid parasites, but they don't really have those as far as I know.
They've got a lot of barnacles on their skin.
Exactly, but those wouldn't really come off. They try to rub themselves on boats sometimes, like to get those off. Just hitting the water, I would think more it's like happiness. Or
I feel now that since you've told me they're intelligent, they're probably there. They have these barnacles all over them, itching them like hell. They can't go to sleep because they're and they're in water all the time, and it's dark and cold. They must feel really depressed.
No, but that's
No.
how
No.
you
It's not so
perceive.
dark.
No,
It's
this
not
That's
is
how
their
you
life. This
perceive
is-
the water. Yeah.
Yeah.
And
may
they
be
also-
they're at
actually
home in
doing-
water. That's
Exactly.
like they would look at us and say, "Oh my God, they're flopping around on land. That must be awful."
Maybe.
Exactly.
Maybe.
Look how dry
Angela-
they look.
Angela, you remember you're talking about looking at a pig and you look in their eyes and you can see intelligence, right? I saw a video of a sperm whale's
Oh,
eye,
yes.
and it just rotated and looked at the camera. Oh my God!
Did you see the Nova? Did you see that Nova special on that?
No, I
Vincent?
didn't. I didn't.
It's called Peter and the Sperm Whale. It's absolutely stunning. You should watch it. It's surreal almost. A female sperm whale actually connected with not a scuba diver even. He was free diving, but the click of his camera and the sperm whale's clicks sort of started to interact, and they recorded all of this, and it was phenomenal. He intercepted them while an entire pod of sperm whales were asleep, and he said it was like visiting another planet. They were all upright in a big giant pod, and then when they woke up, they all started to rub each other. All of them. They were communicating by rubbing, and he says, those whales are trying to teach me something, and it might take me my whole life to find out what that is, or maybe I will never do that. But and then at one point, she left her baby with this guy, and she dove down to get some food, which is a mile deep, and she came back to the exact same spot, picked up her baby, and moved off. It was quite remarkable.
Left it in daycare. That
Dixon.
thing with the clicks is
Yeah.
actually the leading theory for the whale ship Essex, which was the inspiration for Moby Dick.
Yeah.
So it was a whaling ship that was attacked by a sperm whale. And at the time it happened, they were in the doldrums, everything was quiet and flat, and the ship's carpenter was working on the ship, pounding nails into one of the whale boats. And the theory is that may have been interpreted by a male sperm whale as a territorial clicking, and so it came and rammed the ship and sank it, and the whole thing has been a huge story and saga.
Not only that, they've also encountered albino sperm whales.
Yes.
So they have seen white whales.
White whales, yes.
So Dixon, these whales, how long can they sleep and not have to breathe again?
About an hour.
Oh, it's a terrible rest.
But sometimes, it depends on
They're
the
built for it.
species.
Penguins get a quarter of a second rest. I'm not kidding. They have little blinking eyes, and they're taking micro sleep little naps. And that's what they've determined. Their brain turns on.
They don't need 8 hours like humans.
But they do. So they do. Maybe not necessarily 8 hours, but all whales, as many people know, they basically can turn off one hemisphere of their brain while the other hemisphere is still in like a low state of functioning. So they're not fully asleep. So if there was some sort of prey nearby, that they could still be awake enough to react. But they do have shorter sleeping intervals. Some even bob close to the surface and can like slightly breathe depending on the species. But if they are underwater, then it's like, about an hour. But some whales can hold their breath for multiple hours. For diving, like for deep diving, if you're like a Syphidus species or the sperm whale, those are the deepest diving. They can hold their breath for a long time.
A very long time. That's right.
Welcome to This Week in Whales.
I have, for years, been trying to figure out how to revive the left half of my brain.
Yeah, right. I was just going to say, the important thing is to remember to wake up the other half when you're done.
I've got a problem with all of it, but that's okay.
Our last paper, or last story news item for today is paper in PLOS One, sex-specific differences in physiological parameters related to SARS-CoV-2 infections among a national cohort. So this is looking at sex as a variable. And so they looked at sex-specific differences in 4 physiological parameters across a COVID-19 SARS-CoV-2 infection. So they gave people a wearable medical device. It measured breathing rate, heart rate variability, and wrist skin temperature. 1,163 participants, and roughly half males and half females. And then they would report daily symptoms and stuff in an app. And they collected serum also from these patients, and they looked at sex-specific differences. And over 1.5 million hours of data were recorded. And during the symptomatic period of infection, men demonstrated larger increases in skin temperature, breathing rate, and heart rate, as well as larger decreases in heart rate variability than women. And that's it. Okay, so differences between males and females.
Not surprising, but good to look at.
Good to confirm.
So basically, men overreact to stress. Is that the
also
idea
not
here?
surprising? I'm joking.
Well, and the importance of this in the context
man
of
cold.
COVID-19, as people probably remember, is that this is this is a virus that tends to do a number on men and maybe a little less so on women.
Yeah, but twofold difference in mortality. Yeah. Okay, thank you, Amy, for that. What did you say, Angela?
I don't remember. I know I jokingly said that people say, when someone has a man cold, that sometimes when men are ill, they exaggerate a little bit more than
Got it.
other sexes.
I'd never heard that before, but that's cool.
We're just wussies. What can I tell you?
All right.
I'm kidding.
All right, we have 2 papers for you today. And the first one is a Cell paper. Underdetected dispersal and extensive local transmission drove the 2022 Mpox epidemic. And this is out of University of Washington in Seattle, the Fred Hutch Cancer Center in Seattle, and the Sorbonne in Paris, and the Riga Institute in Belgium. So the first author is Miguel Paredes, and the last author is Trevor Bedford, who some of you may remember. He was on TWiV. I guess it was last summer. Yeah. Talking about, he pioneered, genomic analysis during the COVID pandemic, and we talked about that. Now he's extended it to Mpox, and it's quite interesting. So just to remind you, before 2022, most cases of Mpox, and we'll let Rich give you the Mpox rundown in just a moment, but most of them occurred in people with a travel history to Nigeria or an exposure to live animals from endemic areas. But in May, an individual with travel history to Nigeria was diagnosed with Mpox in the UK. And then after that, the number of cases without a travel history began to increase rapidly. Epidemic human-to-human spread. And as of July 2023, 88,500 cases of Mpox worldwide.
And this is the virus formerly known as monkeypox.
That's right. You want to tell us a little bit about it, Rich?
Sure, a little bit. I mean, it is a pox virus, an orthopox virus, in fact, to get a little more precise, in the same group. Its most notorious cousin is smallpox or smallpox virus or variola. The vaccine that was used to eradicate smallpox was vaccinia, also a closely related virus. I think one of the reasons they changed it from monkeypox to Mpox is because monkeypox leaves the false impression that it actually lives in monkeys, when in fact it probably lives in rodents. And is endemic in a couple of different regions in Africa, as we've already said. 2 clades, 1 and 2. One is disease-wise a nastier version than 2. It probably, as I think I already said, is endemic in, or lives, the reservoir host is some sort of rodent. Hang on, I have a...
Is that your cat, Rich?
Yeah, that's my cat, and the cat wants to get out.
I was wondering if it was one of mine.
I thought it was Alan. I was like, is it Alan's cat?
Yeah, and he's trapped in here with me because there is an individual spraying smelly stuff all over the house who has a terrifying vacuum machine.
Oh,
okay?
the monster vacuum.
Yeah, and if he gets out there, he's going to have a bad experience, but he doesn't know that yet.
Oh my gosh!
So
animals.
We'll just put up with him for the time being. He's flopped down again. I think he'll be all right. At any rate, okay. So, and there are occasional zoonoses where there's spillover from the rodents into humans. There can be, although it's not very efficient, human-to-human transmission. There's a certain mortality associated with that, between 1% and 10%, depending on the clade you're talking about. But because of the low transmission, the rodent-specific rodent reservoir, and et cetera, it hasn't been all that much of a big deal globally, okay? And even in Africa, I don't think it's that big a deal relative to lots of the other problems that they're dealing with.
Did Rich, before this outbreak, was there human-to-human spread, or?
Yeah.
It was.
But not well. I mean, my understanding was that the human-to-human spread before this outbreak was by, direct contact or respiratory droplets or something like that. Typically within families, a kid would be messing with a rodent and get sick and bring it home, and the other family members would get it. However, with this that we see now, wouldn't surprise me at all, as if you could look at the whole thing retrospectively, you might identify some sexual transmission. At any rate, there was a case a travel-associated case in Western Europe at one point, and others in the UK that got into the population of individuals, specifically men who have sex with men, and spread sexually within that community. This was, I get the clades. It's easy to get the clades mixed up. Clade 2. Okay, and as we'll see here in the introduction, spread globally, totaling something like 86,000 cases, and so that was a big spill out, if you like, from its normal home into the global population. And the question is, what are the drivers? And this addresses some of that. I should say one other thing, and that is that because it's an orthopox virus, the same vaccines that are effective against smallpox should be effective against monkeypox as well, and have been shown to be so. And we have stocks of those vaccines around, so that they're relatively readily available and distributable. There's also a drug called, I can't think of it other than ST246, but it's
tecovirimat. No, that's
tecovirimat is it also known as TPOX, I think.
TPOX.
That's a very effective drug that I believe should be, and I believe is as effective against Mpox as it is against smallpox and other orthopox viruses.
And both of those things were rolled out in response to this.
Yes.
Yep.
Epidemic.
And actually, another interesting thing here, we're talking about a fairly large, it used to be the largest, now it's a fairly large double-stranded DNA-containing virus. These things are supposed to be, relative to RNA viruses, relatively genetically stable. However, there's a fair number of semi-essential or non-essential genes in the virus, so there's opportunity for genetic drift, and you can take advantage of that drift to track the virus as it moves.
So, Rich, I have a question about the nomenclature then, because you said that, I'm not sure, I don't think you mentioned this specifically, but the name monkeypox, if it normally circulates within rodents, then it's just like the primary reservoir non-human primates that then spilled over into rodents when it was first detected, or why is the name?
No, I think when it was first detected, it was first detected in monkeys.
Yeah.
Okay.
Oh, okay. Yes.
And I think it can spill over from rodents into monkeys in Africa and wind up. They can be an intermediate vector, basically, and it can make its way into humans through that vector, because monkeys get handled by humans in Africa, among other things. So I think the original they exaggerate. The initial associations were with monkeys. That's where it got its name, but it doesn't. In the real scheme of things, it's really a peripheral host.
Okay.
The WHO, when they renamed it, they said the old name could be construed as discriminatory and racist. That's why they renamed it. But I agree that it's probably best.
it's just inaccurate because it's not primarily a virus of monkeys.
I think that's the best reason, I guess. I don't know. But M could be, I don't know. What should it be? It shouldn't be mousepox, because that's a different virus, right?
No. I mean,
it's M.
And you probably don't want to name it after a region.
No, oh gosh.
Okay.
No.
How about rodent pox? How about rodent pox?
Rocks.
That opens up a can of worms. Actually, I should know but I don't know whether there is a specific rodent reservoir associated with this. My guess is that there's more than one, but I'm not sure.
Yeah.
I think probably the rationale of Mpox was just take the monkey out of it, but keep the M so people know what virus you're talking about.
Yeah, that works for me. I mean,
Simple
I have
enough.
trouble. I have a little trouble with Mpox because I've been doing monkeypox for 40 years. But you're right, Alan. That it does work.
All right. So this 2022 outbreak, lots of human-to-human spread outside of endemic areas. So mainly in men who have sex with men. As Rich said, it's milder illness than clade 1. Long incubation period, 5 to 21 days. And they say maybe that facilitated undetected spread, which we don't know about. And it's one of the questions they want to address in this paper. They also want to address whether travel contributed to it and whether vaccination campaigns were effective at controlling it. And so what they do here is use genomic epidemiology. They combine genome sequencing with standard epidemiological information, like, where was the virus isolated, from what person, when, and so forth. And they can make, they use mathematical approaches to answer some of these questions. It's quite interesting.
I want to, at the outset here, introduce, now I'm going off, this is me talking, okay? Going off the edge here a little bit.
I didn't think it was Dixon.
I want to introduce a qualification or some nuance to the word undetected in spread, okay? Because I don't think of these viruses as having a significant amount of asymptomatic transmission.
I was just about to ask that.
Now, in this particular case, I may be overstating that, and I'm not entirely sure. But certainly, with most orthopox viruses, I know there's very little asymptomatic transmission. And so, when we're talking about undetected, I think we're talking about undetected at the public health level. That includes not only undetected but unreported. Okay,
Unreported, that's right.
I agree.
So that qualification.
Yeah.
Okay. Yeah, I agree with that. I think it should be unreported. Yeah. And that's why smallpox was eradicable, because it.
Absolutely. And that's one of the things that makes me think about it.
Yeah.
Smallpox, you only got once. If you got it, you were scarred, so you could tell if somebody had it. Okay.
You knew when you had it.
You knew when you had it. So this is important because some of the take-home messages from this paper can be specific to this particular virus. Okay. Or viruses that behave like this one, okay? There can be viruses with different behaviors that are going to have a different dynamic than this.
All right, so first they describe the outbreak here. First detection in the UK, May 7th, 2022, and then in early May, cases in Western, Southern, Central Europe, peaks around mid-July. In mid-May, cases begin to be reported in North America. And then after that, they start to rise in South America as well. So they collected genomic sequences. They have 1,004 publicly available Mpox virus genome sequences. They have case counts. They know where each of these cases are. And they try and do phylogenetics to see where the ancestor is. And from that, they infer the most recent common ancestor of the epidemic existed between March 9th and March 27th. So remember, the outbreak is first detected in May, but apparently the ancestor virus is circulating back in March. And they think this was in Western Europe. Phylogeographic estimates put it in Western Europe, because you know where each of the genome sequences came from, right? So you can do this sort of calculation.
So define Western Europe. There was a pod in Portugal,
without starting
right?
a war.
Portugal
Exactly.
and Spain.
And Spain and Italy a little bit, but Portugal and Spain, it looks like.
So getting back to the symptomatic versus asymptomatic, it's hard for me to imagine somebody who has obvious pox, regardless of the cause, not knowing that this was transmitted by a certain behavior, and to continue that behavior would be unethical. Do you think that's not a problem here?
In
other words,
this gets into a sensitive area.
Well, I'm trying to not
because
lift up that rock, but almost like a red badge of courage.
No, not a red badge of courage, but we may be talking about a situation where it's easy to ignore stuff like this.
Yes.
Okay, and press on with your behavior.
Well, another question would be then, how you said it takes 2 weeks to 3 weeks. This one has a longer incubation period. What is the transmission period? Same as the incubation period or different?
I don't know. You mean how long can it be transmitted from an individual to another?
Before it becomes symptomatic.
Before. Well, that's just it. I'm not sure there is much asymptomatic transmission. Okay. Though it could be that there are, in this particular case, symptoms that are subtle enough so that you could wind up transmitting it symptomatically without really understanding what's going on.
Okay.
The presentation of this disease, I don't think, is as in all cases as dramatic as some other orthopoxvirus diseases. I think of poxviruses infections as really frank, discrete, annoying, painful, itchy lesions. Okay, pustules. Yep. But in this case, some of the symptoms are described as rash. Okay. So it may be more, I'm not intimately familiar with this, but I imagine that there can be more subtle symptoms where you could be symptomatic, but not so frankly symptomatic that you really paid a whole lot of attention to it.
Or you could confuse it with like an allergy or something, like contact dermatitis, or who knows. I'm also not very aware of it. Right. But if it's a rash as opposed to an itchy pustule, then it could be.
Right. What we're going to, spoiler alert, what we're going to see from this paper is that when the consciousness of this, the cat's been released. When the when the consciousness of this elevated, it took care of it.
Yes.
Okay. So that's a clue, really, that once you understand what's going on, you can take appropriate action, which implies to me that what we're saying is true. There wasn't a whole lot of asymptomatic transmission, but it's more of like, I've got these symptoms. This could be this. I'd better chill out here. Okay.
Right. Nobody was thinking of Mpox. Yeah. And you got a little rash or something. Whatever. I mean, skin gets itchy sometimes. But then Mpox was the big story, and now all of a sudden, my skin's a little itchy. I probably ought to get that looked at.
Yep, exactly right.
Yeah.
All right, so they also look at these data. They find examples where single introductions result in big local spread. So local spread played quite a role in these regional outbreaks. And in fact, rapid early spread in Western Europe led to introductions in other global regions. And then for North America, they had repeated dissemination into North America more than once, in other words, and then subsequent sustained community transmission. So community transmission is a word for spread when you don't know you're spreading something. You don't feel badly enough to suspect that you're sick and you're out and about, and that's community transmission.
Just spreading in the community, yep.
They then go on to look at within region transmission dynamics. So far we've had a global picture, right? And they spend a lot of time talking about their methods, but it's very interesting. It turns out they're computationally intensive, right? They used a couple of different methods, and they say for certain approaches, it took 25 and a half days of computational demand for one thing and 34.3 days for another. And they said, that's too long. And so we had to make efforts to reduce that. Otherwise, this paper would take forever to get out.
And also, CPU time is like billed by the second. So,
yeah. Imagine, my job is 34 days. Yeah. Everybody else would kill you.
All right. Anyway, when they finally do this, they see evidence of viral circulation before detection in each global region. And they find the largest outbreak clusters arise from introductions before detection by public health surveillance. And then introductions after detection are typically a single case and they get extinguished. So as you're just saying, when you're not aware that something's going around, like a rash-inducing virus, that's how you spread it. And then once people are aware, Mpox is a thing, then it goes down. And they say that's really evident in their data. Let's see. So then they said, is there a difference between our data and case counts? And they find divergence in the early months of the outbreak, which they say indicates underdetection of cases in the earliest months. As we were taught, now they use the word underdetection instead of asymptomatic, which is really good. So that's May, June, etc. But by August, there's concordance between their data and the epidemiological data. So there's no more underreporting. And again, that's when the word
awareness.
is out.
Right. The awareness goes up.
Right.
Yep.
So then they look at what happens after initial introduction of virus into a region. So they analyze transmission chains resulting from introductions. They see a bimodal pattern where most introductions give you a single imported case, and a very small number of introductions give you explosive and widespread local transmission. Early introductions give you larger and more persistent transmission change, where, again, those introductions that happen after public health detection give you smaller outbreaks that went out quite faster. And they find that looking at air passenger volumes, they're a good positive predictor of migration between each region. So travel does make a big difference here and drives it.
Well, travel drives it partly. It drives the initial introductions.
Right.
That's right. Yes,
But
right. It has to come from.
Yeah. But then
to get from Spain to the U.S., you got to get on a plane.
Right. So for it to travel, it does have to get on the plane. But then air travel, if air travel had been shut down after the introduction, that would not have shut down the epidemic because you've got local community spread. So it really argues against travel restrictions.
Yeah, the same thing with COVID, right? In the early months, they say, let's shut down travel from China.
The horses out of the barn.
Too late.
Yeah, I wonder, that's a really interesting finding, and I wonder to what extent that applies to other pandemic viruses, because if that is routinely or globally applicable, almost regardless of what pandemic we're talking about, that has a huge impact on how we deal with it. I think one of the ongoing lessons from the COVID pandemic is this trying to understand on a global scale the best way to deal with this, okay? In particular, trying to balance public health measures with social and economic considerations. And air travel is one of those things. And it could be that with a lot of these things, by the time you know what's going on, okay, and want to restrict spread by restricting travel, it's too late.
I am going to predict that will be a general rule, and my reasoning is that if you have a virus that needs multiple introductions in order to continue spreading, then it's not going to be able to be a pandemic anyway because it's not going to be spreading enough.
Yeah.
What makes a virus capable of really spreading around the world is that it's highly contagious. And once you introduce something highly contagious into a population, you're done. There's no point shutting down travel because it's already in.
Yeah.
And all you have to do, man,
somebody could model that, I think.
All you have to do is boot up Flight Tracker 24 and look at the planes that are buzzing around the globe at any given instant, and you realize that anything that breaks out somewhere is going to be global almost instantaneously.
Yeah, it's a frightening graphic.
They wanted to know the contribution of introduction versus local spread in these regional outbreaks, and they see that there's usually just one introduction and then extensive local spread. You don't need multiple introductions to have a local epidemic. So following the initial seeding, local transmission dominates, and introductions play a limited role in the later stages of a epidemic. I guess that's kind of intuitive, but you have to find out. All right, then they looked at transmission dynamics, and they calculate RT. RT, right? This is the what's the name of this? Reproductive number, right? R,
Right.
which is some measure of how many people an infected person on average can infect. And RT is the time varying reproduction number, as opposed to the R naught, which is supposedly a constant but really is not. So they observe high RT when virus first gets established in each region, and then it decreases to less than 1, and the epidemic stops, because you can no longer sustain it if you have one person infecting less than one other person, right? They remove, when they do their calculation, they can remove the introductions, and it has no impact on the spread. And that's consistent with what they just concluded before. And what I found interesting, they say so this RT is calculated using a mixture of case counts and their genomic data. If they only use case counts, which is what most epidemiologists do, they vastly overestimate the transmissibility of Mpox for each region. And all I want to do is remind you of, with each successive SARS-CoV-2 variant, the R naught or RT kept going up and up and up. And I think that's possibly why they weren't using a combination of the data. Okay, U.S. vaccine campaigns. What did they do? In North America had most cases throughout the epidemic, so they did a lot of analysis of that. Introductions only account for 5 to 15% of local spread, which is consistent with what we've already heard concluded here. So preventing introductions, as we said, by travel restrictions would not have made any difference. And then they look at the RT for North America, and it begins to decline before we start vaccination. Okay? Eventually, vaccination does have a big role in stopping this epidemic, but the decline begins already before you start vaccinating. And they look at the numbers of people vaccinated, and it's very low by the time the RT drops below 1. So the other measures that we've talked about, right? Public awareness, that is what started to bring it down.
Now, I would comment that this is a situation where public awareness can have a huge impact on the spread of the infection. There are other infections where public awareness, may be important, but it can't have as big an impact, like SARS-CoV-2.
I was just going to say.
Where you're talking about at least half of the infections that are spread asymptomatically, okay? And so in those cases, vaccination is going to have a bigger impact because, you can't snuff it out just with awareness and behavior.
How about when you
being
treat
a respiratory
somebody,
virus.
though, Rich?
Excuse me?
When someone is being treated for this, though, how long after they start treatment are they not infectious?
I can't tell you specifically. I would guess a week or two. Okay, I think of this as being transmissible as long as you have active lesions.
Okay.
And if you start treatment, it's going to take at least a week, and possibly more like 2 weeks for those to actually heal up. Because
saliva and other secretions don't have. It's only specifically the pustules that are full of viral particles, or is that
in this case, I'm not entirely sure. Certainly, the pustules are going to have live virus in them. I'm not really. I think there may be some systemic spread, which would mean that ultimately you could potentially get some respiratory spread, but I don't think it's a big deal in this case. In the case of variola, That was everything. The primary spread was through respiratory droplets. It was the primary infection was usually a respiratory disease. Then it would spread systemically, and you would generate pustules. And now you got a big problem because it can be spread from that individual as a respiratory infection, from the pustules, and from things that person contacts, like sheets and blankets and clothing and that kind of stuff that get virus on them from the pustules. This, I think, is more confined to direct contact from the lesions.
Okay.
All right, so the last point I want to make on this is that they're looking at their data. They notice that a few introductions give them a lot of sustained local transmission, and then those make few downstream infections. And that's consistent with some people being more responsible for transmission than others. And this can be measured by a parameter called the dispersion parameter, or K. It quantifies this heterogeneity in transmission. In other words, some people transmit a lot and others transmit less. And so so the low values of K correspond, counterintuitively to a higher degree of heterogeneity in transmission. And when this transmission heterogeneity is high, maybe you want to target those individuals that could have an impact, right, on the outcome. So they find by looking at this for the Mpox outbreak that there's a great deal of transmission heterogeneity. There are people who seem to transmit to a lot of others, and then others who don't.
But that's also based on behavior, you'd think,
yes.
Because with SARS-CoV-2, obviously it's a respiratory pathogen. If you're on
that's right.
like subway, you're going to be, or in a classroom or anything. But if you're, obviously it's not only via like sexual contact, or if you're literally
Close
rubbing
contact.
against people, some sort of contact from skin to skin. And if you're contacting 50 people as opposed to 10, then that would make sense that it would be so heterogeneous. Like some people
And
might
if you've
not
read
come in
my
contact.
pick from a few weeks ago, Strange Bedfellows by Ina Park, you will have read. Yeah. So the term she applies to there are people in a sexual network who will spread to more people than others, and she refers to them as being affectionate and popular.
That's right. That's what they say here.
And going back to Angela's other question about transmission, I think if there were, in this case, if there were significant systemic spread in an individual and respiratory transmission, it would have had a much different profile in terms of both the epidemiology and what it looks like. It's clearly, and not only that, but the symptoms are localized primarily to areas associated with sexual contact. Okay. So this is a direct contact, sexually transmitted disease.
Okay.
So do the lesions usually show up at the same point where the inoculation occurred?
I believe so.
Okay.
Yeah, I think that's correct. Yeah. But there is some spread, so that we used to talk about that and try and figure out. Sometimes you had long distance spread, and it's not clear what's going on there.
Right.
But even with SARS-CoV-2, if you had someone who's infected, asymptomatically infected, and goes into a bar with a lot of people, that can be a super spreader event.
Oh yeah,
right?
Of course, yeah.
So in a sexual network, it's sex, but as Angela said, in a community transmitted,
it's breathing.
It's breathing. You're in a room with a lot of people, you're going to infect them.
Okay, I'll stop breathing.
You're picking on Dixon again.
Always tried. All right, so that's the paper. I just want to mention one more thing here. They talk about the evolutionary rate, the mutation rate of these viruses, and these clade 2B Mpox, they say has a significantly faster evolutionary rate during human-to-human transition, driven by editing by a deaminase called ApoBec3, which deaminates bases and changes them in the genome. So that's a cellular enzyme that do that. And they say, consequently, this mutation rate is close to that of an RNA virus, which is very interesting. Because DNA viruses typically, we think and know sometimes that they have low mutation rates, but this one is pretty high. So I thought that was interesting.
It was comparing it to variola, which was 1 to 2 substitutions per genome per year, versus this Mpox
16.
is 16.6. That's crazy, the difference.
Throughout this, I found myself thinking about our TWiV episode with Mike Merschlinsky, okay, who really is at least in my sphere of influence, the monkeypox expert. And his bottom line at the end of that episode, when we said, okay, so what's going to happen? What do we do about this? And he just said, behavior. Okay. And that turns out to be true. Awareness of what's going on and appropriate behavioral adjustments, and it took care of it.
And I would also want to point out that this may have had a lot to do with the specific community this affected.
Yeah.
So men who have sex with men tend to be highly aware of viral spread and viral dynamics, and because of the whole HIV thing, they're more likely to be able to grasp this and say, "Oh, all right, got to do precautions." So
the lessons here. Public health awareness is really important. Vaccination didn't start the decline here. It was public health awareness. And behavioral modification is really important. Education campaigns. But they also say, because we didn't detect these early infections, we need better detection. We need better surveillance and detection.
Yeah. And that does apply to anything.
To everything.
Okay? Yeah. The earlier we can get on top of it, and we've been saying this for a long time, that the earlier we can get on top of it, the better off we are.
The delay in ability to test in the COVID-19 pandemic was a massive problem, and if you'd had quicker tests for this, but I'm on the other side of that. You can't test every patient for everything because that's just a huge waste of resources. But you need to have something that's quick and
quite often, I'm sure that in the beginning of this, people saw this and didn't understand what they were looking at.
Yeah.
Okay. And there's going to be other diseases that come along where people see it and they have no way to think about it at all. It's entirely novel. What is this?
And if you're interested, go read the limitations of this study. They start by saying, our study has noteworthy limitations.
Yeah.
And then they make note of them.
They make note of them, yes. And they say mostly they don't matter. All right, onwards. This is a Nature Communications article, a humanized mouse model for adeno-associated viral gene therapy. Let's see, the first author, is there co-first authors? I have no idea. These authors contributed equally. Mercedes Barzi and Tong Chen, the first two authors, and then the last author is Karl Dimitar Bissig. This comes from Duke University, Baylor College of Medicine, Texas Children's Hospital, Duke. Yeah, a lot of Duke in Texas. So this is all about using adeno-associated viruses as vectors. Rich, what's an adeno-associated virus?
Since Kathy's not here.
I was literally going to say we have to explain what this is. I added a little video, actually, a 3-minute YouTube video to our sheet that maybe we can post for the listeners. But I'm sure
and that's good. Job. And let's not forget to come back to that, because I have a caveat for that video that I became aware of because of this paper. Okay, so adeno-associated virus is called adeno-associated virus because it was first detected as a virus that showed up in cultures of adenovirus. Okay, and in fact, so it is a small single-stranded DNA virus in a naked capsid, and by small I mean teeny weeny. We're talking about, I think it may be as much as 5 kb, but I think it's about 5 kb. Has just two genes, rep and cap. Rep is required for genome replication just to get it rolling, okay? And actually it's required at a couple of different phases in the replication, but mostly cellular machinery is used. Cap is the capsid protein. Some of these things can replicate by themselves. Some of them are so lame that they need a helper virus, like, for example, adenovirus, okay, to provide functions that they are lacking.
Now you could argue they're clever not to have a lot of
Yes,
baggage.
That's true.
Right. Get by with a little help from my friends, right? So over the years, this thing has been engineered into one of the prime gene therapy vectors, where what is done is to basically gut it of all of the functional protein encoding genes in the virus, leaving it just with some bits of DNA on the ends that are important for packaging and replication of the genome once it gets into cells, and it becomes, and that way the engineered virus becomes just really a delivery vehicle for these genes into cells. And you use various techniques to get the engineered genome to be replicated and packaged. You make a stock of this, and then you can introduce it into a subject, and it infects appropriate cells. And the DNA is maintained. The dogma is that the DNA is maintained as an episome, sometimes concatameric. That is an extra chromosomal molecule and can express genes over a prolonged period of time. It's a parvovirus is the larger realm of these viruses. And there are human adeno-associated type parvoviruses, and there are AAV type viruses of other creatures as well. There are many serotypes in humans, some more common, some less common. This becomes important in trying to engineer the virus because you want to try and avoid any innate pre-existing immunity to whatever vector you're using. So you try and use a rare serotype. You can also use the different serotypes to help you target specific tissues and et cetera. The one other, I may as well introduce the caveat now. There's one paper that's referenced in here that I was unaware of. That's where they use a primate model infected with or treated with adeno-associated virus, and they noticed that gene expression is robust in the first few months after introduction and then falls off to some minimum. And their explanation from that, based on their data, is that you get a lot of expression from the episomal form of this thing early on, and then there's some integration in a small number of cells, and you get a trickle of expression beyond that. Now, that's in a primate model, so we don't know if that happens in humans, but I was entertained to see your video, Angela, because that goes against the dogma that I've been taught over the last several years. And if integration actually happens at any significant level in humans, that's going to be an issue for gene therapy.
Yes.
And we'll see what happens.
Yeah, and so people have been working on this as a gene therapy vector for, as Rich said, a long time. And it has actually been successful. In at least a couple of cases, it's had its ups and downs, but there are now 2 FDA-approved therapies for 2 different types of hemophilia, Hemgenix and Roctavian.
Isn't that an RSV something?
Sorry, I just love that. So this is actually something that is in use now clinically and could potentially be used to treat many other genetic conditions.
And there haven't been problems that we would associate with integration at this point. The other one thing that's relevant to this paper that's worthwhile is a lot of these therapies can be implemented by infecting hepatocytes, cells in the liver.
Right.
And that is a target of many of the serotypes. Okay, so that's relevant to this paper.
There's also an AAV-approved, FDA-approved vector for blindness, for a certain form of blindness, Leber's congenital blindness, and it's called Luxturna.
Right.
I think it's $500,000 per eye.
Oh.
Yeah, and now you can see.
Yeah,
you can see.
That's a big deal.
How much
Yeah.
is it worth to see?
How much is it worth?
That is. Yeah.
So the issue in this paper is to try and make a better animal model, right? Most people use mice for preclinical studies, right? Before you put it in humans, you want to put it in some kind of animal, and mice are typically used. They mentioned, maybe non-human primates would be better, but this is expensive, and ethically we don't want to do that. So the problem with mice is that these AAVs infect the liver really well.
Mice exaggerate.
Mice exaggerate. They absolutely exaggerate. Now, one of the things that they have done to try and get around this is they put human liver, they make human liver chimeric mice, right? So they add pieces of human liver to the mice and they grow and you get a chimeric animal, like about 60% human, 40% mouse liver. And you can then say, does my vector infect the human liver cells? But the viruses still prefer to infect or preferentially infect the mouse liver cells. And they say this is like a sink for your virus.
Great use of the word.
This is not good. And it confounds the finding. So what they wanted to do here is get around that problem. And what they did was, so I think we actually did this paper a while ago on TWiV, identification of a universal adeno-associated virus receptor. All the serotypes bind this receptor for cell entry, AAVR. And they just knock out the gene in mice. Apparently, it's not needed for anything we can tell in mice.
They haven't taken them to the opera.
They can't tell Wagner from Mozart anymore.
And so they combine this knockout mouse with a human chimera. And so now no mouse cell has an AAV receptor. And if you stick human cells in the mouse, the virus is going to reproduce in the human cells, right? So that's the basis for this paper. And so the mice they use are not your normal lab mice. They're actually knockouts for a couple of genes that are quite significant in giving you immune responses. There's the IL-2 common gamma chain. There's the RAG2 gene.
Yeah, there's no T cells or B cells in these mice. And the FAH was something to do with hepatocytes, wasn't it? Was it kidney and hepatic? I tried to look up the gene, the FAH knockout, and it just said that it was associated with.
I couldn't figure it out what it does, but
I assume that those knocked out genes are there or not there, so that the mouse liver will accept the human graft.
Yes, exactly.
Yeah, exactly. The lymphocytes for sure. Yeah, if they were not lymphopenic, the human graft would not.
Yeah, they would reject the human cells if they didn't have. I mean, I think these mice are very. So you said T and B cells, right, and NK cells probably too.
And then RAG2, I believe so. Yeah, for sure B and T. Let me see. NK.
So that's why they take the human liver cell transplant. So they take this knockout, they actually make the knockout AAV receptor mice using CRISPR, and they then put that on a background of this IL-2, RAG, FA, et cetera. So they're called TIRFA mice, TIRFA, TIRFA, as opposed to TIRF. Maybe it should be TIRF, TIRF versus TIRFA, with or without the receptor. And these mice seem to be okay. They have normal liver functions, no pathology.
They're okay for mice with no immune system.
Yeah, right.
So to clarify, RAG2 has actually increased natural killer cells. I just looked, so it's only B and T. But they have like this compensatory mechanism of increased NK cells.
But the IL-2 receptor gamma chain is another, that's like SCID, right? IL-2.
I believe so. IL-2 is important for T cells specifically. The IL-2 receptor for B cells? I actually don't know.
I'm getting boxing things when I do IL-2 R gamma knockout.
Oh, knockout.
It's mostly T cells, as far as I know.
What is the deficiency?
I'll call Brienne.
Yeah. It's got absence of mature T cells.
Mm-hmm.
It's basically the defect in X-linked severe common immunodeficiency.
So it's
people.
SCID.
So you have mature T cells, decreased NK cells, but you get an increased or normal number of B cells. But the B cells are taken care of by by the
Exactly.
rag. Yeah.
Yes.
And if they're also with the IL-2R knockout, then they're also decreased NK cells, which you wouldn't have with the rag-2. So I guess they're compensating for all of the
right.
immune compartments.
All right. Well, they made these mice, and now they bring them to the lab and test them. They infect them with AAV8, which is a commonly used serotype. They have a reporter gene in so they can easily track it. They have the tomato reporter. You can
V8
guess
is a
Tomato.
tomato juice.
V8 is a tomato juice stick.
It
so
is.
Very good.
Yeah.
It is. So they inject intravenously into both TERF and TERF-A mice, and minimal transduction in most organs of the receptor knockout, which is what exactly you would expect because the receptor is gone. And transduction, they're looking at, the red fluorescence or signal in these mice. Except heart and liver seem to still take up and virus and produce some fluorescence. So maybe something else is allowing entry into those tissues. I don't know. Could be. And then your TERF-A mouse had only production of the reporter in human and not mouse liver. So very nice sections where you can see the mouse tissues are dark and the human cells are red.
Mm-hmm.
So this virus, so you put human cells in, and of course the human cells have the AAV receptor, right? It's the mouse that lacks the receptor, and now the virus can infect the human cells. So you can study whatever you've got in your AAV, you can now put it into these animals and study what it does, right?
Presumably not a tomato gene for clinical use.
No, you don't want to put a tomato gene in. Well, yeah, exactly right.
So to me, the really critical image for my poor brain in understanding the data was in Figure 1G, the last two panels where they're doing immunofluorescence on the mice. And it took me forever to sort of figure this out. But the bottom line is, if you have any cell that's infected, so the human cells are green and the AAV infected cells are red. If you have a human cell infected with AAV, it's going to be yellow.
Right.
Okay?
Like the Ziploc. Yeah.
And so if there are any mouse cells that are infected with AAV, they're going to be red. Okay? And if there's any uninfected human cells, there's going to be green. And what you see is that in the parent, if you like, mouse that doesn't have the receptor knockout, there's a whole slug of red cells and very few yellow cells. So that's a whole bunch of mouse cells getting infected by AAV and very few human cells. In the one where they have the receptor knockout, all the cells that are not green are yellow. Okay, so there's none that you can see that are mouse cells infected with AAV. So it's brilliant, just brilliant, and absolutely clear once you can wrap your head around it.
Yes.
It's good. I'm so happy, Rich, you said this because it took me like 10 minutes as well. I was looking at it and I was so confused and I was like, I'm just stupid. I don't know what this is.
Well, wait till you get to the next figure where they don't even tell you the results.
Yeah.
Actually, I was reading a paper and trying to look at the figures, okay? And I look at the figures and I couldn't understand it and I was running out of time. So I said, dude, I'll just finish reading a paper so they understand what they say here. And then if I have time, I'll go back and see if I can figure out whether the data support the conclusion. And that's when I figured out that little 2-panel thing. And I said, okay, that's enough for me.
No, that's very nice. That's very good. All right, so that's the big finding of this paper, that you can limit the transduction to the introduced human cells. They do some other experiments. We'll go through quickly. For example, they say, okay, where's the viral DNA and RNA? So they can do in situ hybridization to detect that in the livers. And they can see lots of AAV DNA and RNA in human hepatocytes, right? So you can tell which ones are human. But they also found DNA in murine hepatocytes of the knockout mice. but not RNA. So the DNA is somehow getting into those cells. But since there's no RNA, I don't think that's a problem for their model. But I guess there's some kind of uptake of DNA, right?
Yeah.
Or particles into the non-receptor bearing cells. All right. And now then they say, what about other human tissues besides liver? Could we use this model to study that? So this is very strange. They do this and then they say it's really not good. They put a teratoma into the mice, right? A teratoma is a tumor that makes tissues from all three germ layers. Probably Angela knows all about these.
This is
right?
actually, so the first time I saw one in vet school, it was terrifying. They normally have like, well, they're very heterogeneous, but they'll have like hair and teeth.
They can have teeth.
And yeah, it's terrifying. I cut into one once and there's like pieces of teeth, but they're not fully formed teeth. They're just like calcified chunks. And some of the hairs were like 5 centimeters long, like chunks of hair. So yes, teratomas are.
So these happen in animals, not humans?
Also in both.
They can happen in
humans
humans.
and animals, yeah.
They do.
They're the
But you were
things
seeing
of,
them in animals, right?
I've seen them in animals. Yes, mine was in a necropsy. But they're the things of nightmares. That's all I can say.
Yeah, so the important thing for this paper is that they have all 3 germ layers.
Germline, yeah.
So you can introduce the teratoma, and it gives you a representation of how you could potentially get this to other tissues.
Yeah, because there are going to be circumstances where you may want to target something other than liver. So you need a model for that.
Yeah.
So they introduce these teratomas into the mice and they let them grow for 3 to 4 months, and then they put the AAV in them. So these are the receptor knockout mice. And then they do sections of the liver, and it's figure 4, and they never tell you what the results are. They stain them with antibodies to different markers to identify different human tissues, right? But they never say what's happening, and you have to figure it out yourself, which is fine. But I think it's weird that they don't tell you the results, right?
Well, this is it's a short paper, and what is this? Nature.
It's Nature Communications, though. Like
Nature Communications.
You'd think they
should have given them enough space to do that.
Yeah,
I would think so. And then they also put human liver cancer tissue into humanized livers, and they show that the virus will reproduce in that as well. So you can introduce a variety of things into these mice, different cell types, and get infection.
But then they turn around and say, yeah, but it's not a great model because the teratomas are not consistent.
Yes.
Because they're heterogeneous. They're all different.
They're heterogeneous, right. Sometimes you get teeth, sometimes you don't.
Exactly.
It's so weird. Yes.
So
this is really good for human liver, targeting AAVs, and other tissues remains to be seen what you're going to do. And that's really it for the story. But I think that, they're very excited. They say this is overcoming significant limitations. And we're looking forward to using it to study AAV and vectors in these tissues. So.
It's amazing. These things take a long time. They take decades and hundreds, if not thousands of scientists to really, and you work out a few kinks and other kinks pop up. Okay. So this is really something.
Yeah. And this is something that would help what the drug industry calls fail early. So many of these gene therapies over the years, it's been, it looks great, and then it gets into humans like it doesn't work.
Yeah.
And this would help really, hopefully, winnow that down and say, it doesn't work so well in mice.
So, Richard, if another kink does arise, we should throw out the term tomato and introduce the term Lola.
Rotten tomatoes.
L-O-L-A.
Anyway, so you could use this for other tissues, right? You would introduce other human tissues in as well. So in theory, you could do more than liver. Mm-hmm. Okay.
And Angela, the video you found really is good. We could.
I thought it was like a little 3-minute video that explained it fairly well. Fairly accessible. And like the animation was very nice.
Yep.
So we can add that to the show notes.
Let's do a couple of email. And I put this first so that Angela could read it because it's to her. Go ahead, Angela.
Okay. Hunter writes, Dr. Mingarelli, I am so pleased that a veterinarian has joined the TWiV team. I have enjoyed your contributions to the podcast despite your having to juggle your PhD work at the same time. I hope you can access this link from the most recent JAVMA. So this is a veterinary medical journal. It is extremely scary and disheartening, as now we are endangering the health and welfare of our pets, in addition to potentially increasing the risk of rabies in people. And then there's a link here, which unfortunately is not open access, but he continues with, this is the part that scared me the most. Results that show that nearly 40% of the responding dog owners believe canine vaccines are unsafe, while more than 20% consider them to be ineffective, and 30% think that they are medically unnecessary. Notably, about 37% of the dog owners surveyed believed that canine vaccination could cause autism in their pets, despite the lack of scientific evidence supporting this claim. As a retired food animal veterinarian, this survey borders on surreal. Oh, how far we, in brackets, scientists, have fallen in the eyes of the general public. Keep up the great work, but I will continue to enjoy my pork. Respectfully, Hunter Lang, Doctor of Veterinary Medicine. Well, if you enjoy the pork, I know it's delicious. I just choose not to eat it. And as for this publication, so I couldn't actually access it because I don't have access to this journal through McGill, through my university. But the fact that it's like not surprising that many people are also now cautious of vaccinating their pets, because just misinformation in general among humans not wanting to vaccinate themselves, they're just extending that to their pets. It's very unfortunate because a lot of the things that we vaccinate for in pets, like rabies, you're just putting your animal's life in danger. So if you have raccoons and squirrels, exactly, and distemper and parvovirus, there's so many other things that are in the environment. They are literally surrounded. Your pet is surrounded by them. And by not vaccinating your pet, you could potentially be shortening your pet's lifespan by a decade.
And with rabies, you're endangering your own life.
Yourself, exactly.
And that of everybody in your neighborhood.
Exactly. Your own family, your pets, because then your pet becomes a vector, and it's very, disheartening to say the least, Hunter. Yeah. Please, everyone, vaccinate your pets, vaccinate yourself. Everyone get vaccinated.
All right, Rich, why don't you take the next one, please?
Sure. Eric writes, and I think I can reveal that this is obviously our friend Eric Delwart, as you'll see, the father of dark matter. Dear Vincent, in TWiV 1091, Rich mentioned my use of the term dark matters to describe the large fraction of sequences of unknown origins found in complex biological samples, such as the gut microbiome or wastewater. Dark matter is a term I shamelessly borrowed from the field of astronomy, which describes the missing mass needed to explain the structure of the universe. Typical metagenomic analyses identify biological species through sequence similarities to already sequenced reference genomes. In the preprint from the Stanford labs that we discussed, this is relevant to that, novel computational tools were developed to identify short circular RNA genomes capable of folding into rod-like structures and encoding previously unknown proteins. A small fraction of the dark matter was therefore illuminated without reference to prior genomes, and revealed the rather common existence of a new type of viroid like obelisk elements. Of course, some bioinformatics-based predictions will have to be tested using wet lab experiments, likely using synthetic genomes. Many more surprises are doubtlessly in store for young computational biologists interested in novel ways of mining the already immense and exponentially growing public sequence databases. Or, as Vincent concluded, seek, S-E-E-K, parenthesis, or rather seek, S-E-Q, and parenthesis, and you shall find. Eric. That's delightful, Eric. I really appreciate that. DNA, is really a problem, right?
Yeah. We may have to use seek and you shall find for a title sometime.
Oh.
That is cute.
Yeah, we
That's
should.
very cute.
That's very good, Eric.
Yeah, it's And I like the plug for bioinformatics for people
Mhm.
looking for something to do with their lives.
Yeah.
Because we're generating, many, 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 many more times data that we know what to do with. Okay. And even when we can see in the dark matter things that we at least vaguely recognize, there are layers of meaning to it that we don't understand. Okay. And bioinformatics is going to address all that.
Well, hopefully.
Some of it, maybe.
Maybe.
That's up to the next generation.
Bioinformatics
You
has
folks
the
out
best
there.
chance of addressing
Yeah.
that. Yeah.
Okay.
Yes, that's right.
Well, you could sell it that way anyway.
Well, I pitch it to my daughter all the time. She
Okay,
likes math,
good.
and I'm like,
All right.
yeah, bioinformatics.
Dixon, can you take the next one, please?
Yes, sir, I can. Bob writes, read the dengue snippet article. I could not read the Nature Medicine article as I do not have a prescription or subscription. I did read the abstract. I wonder if there was some selection bias in this study, specifically folks who are not very sick did not go to a hospital. Well, I never saw dengue in my middle American practice. I think the symptomatic treatment of mild dengue would be outpatient acetaminophen or NSAID, rest and fluids. Just wondering if they only picked the more ill patients inadvertently. I have no opinion about this whatsoever.
Well, so this is the paper where I think it was from India, where they found that the first dengue infections could be severe, right? Because we're always thinking
I
it's
see.
the second ones that are severe.
Sure.
And so it doesn't matter actually if there was selection bias because you're getting severe cases in a first infection, and that's the point, right?
Right.
Yeah.
Even if you missed some patients, you're getting severe dengue, and these people had their first infection. So that's the conclusion. I think it's okay, Bob, as far as I can tell. All right. What do we have? We have Alan. You can take the next one.
Kurt writes, hi, listening now for almost 3 years. I've donated, but other than that small gratitude, there are not words in English to acknowledge what all of you are providing. So I'll try a little Russian. Bolshoy spasibo. Now, in episode 1084, you mentioned that 20% of cancers are caused by a virus. Is this 20% of cases or 20% of known cancers? That could make a colossal difference and impact further research and its clear value. Gratefully yours, Kurt. I think that's 20% of known cancers.
Yes,
isn't
that's
it?
the way. That's the way I understand it. And I've in my previous life actually, touted this number along with a slide that enumerated lots of different cancers, different types of cancers, and identified those which were caused by viruses.
So one, for example, that comes to mind is EBV, and I think gastroadenocarcinoma, Epstein-Barr virus. And that's like the number one that I always think about, which is funny. Other people think of, I guess, does everyone have one we can highlight?
Well,
So
EBV and cervical cancer.
Yeah.
Also
EBV
cervical,
causes a
sure,
bunch
EBV and
of-
everything, actually.
Yeah, EBV does
Apart
a bunch.
from
Then we have
EBV.
human papillomaviruses, right?
Hepatitis B virus
Hepatitis
gets
C.
in there.
Hep C, Hep
yeah.
B, HIV, of course. And there are others that
What
we're missing.
cancer is associated with HIV? That I actually don't know.
Yeah, well, they're not. It's not an oncogene-driven
Oh.
thing, right? But it's from the the constant proliferation of various cell types in the light of immune inflammation, right?
And
Yeah,
you're limiting this just to human, or
yeah, this is human cancer.
Oh, yeah,
Just
it gets out of
humans.
control if we, If
So
we
we're
open
just
it up to the
talking that
cetaceans, Angela would be here
People,
for
yeah.
hours.
But people with HIV then are more susceptible to different types of cancers, what you're saying.
Yeah, they
Not
get
that there's
different
one
types.
associated. Exactly. Okay, so.
Yeah. I mean, they will get EBV cancers, they'll get
Mm-hmm.
HPV cancers and but also non-viral cancers as well, right?
Okay.
Kaposi sarcoma, is that falling into that group?
I believe so.
That's
That
fine.
was
as
another
well.
herpes
Is it?
virus.
Yeah.
Yep.
Yeah, Kaposi sarcoma, herpes virus, right?
That's also in animals, in case. you get-
Interesting. All right, let's do one more here from Daniel. Daniel writes, Dear TWiV, in episode 1091, Skeeter Poo and Obelisks 2, Rich said that you can't do phylogenies just with structures. And Alan mentioned that you could use morphogenic techniques to solve that problem. Alan is very much correct there. In fact, Scientists have been using such techniques for a few years now. As such, I thought this would be the perfect moment to talk about the blossoming field of structural phylogeny. I'm not a structural phylogeneticist, but I'm doing a PhD in structural biology and attended a meeting on the topic a few months back purely out of interest. Structural phylogeny has been taking off in recent years with the advent of more powerful computational tools and predictive tools like AlphaFold. A range of different algorithms are available for computing pairwise differences between structures, and these are used to assemble distant matrices for phylogeny construction. Experimental structures are, of course, favored. However, deep learning-generated structures can also be used to supplement these databases. The most powerful aspect of structure-based phylogenies is their ability to probe deeper in time than sequence-based phylogeny. Specifically, while sequence-based homology in distantly related proteins is lost over time, structural homology can be detected in proteins that separated billions of years ago. Genetic sequences mutate and amino acids are changed, but the actual base structure of a protein is usually maintained. This is comparable to how all vertebrates have the same body plan, which has been modified over time to give a huge diversity of vertebrate organisms. Similarly, modifications to a protein fold can give rise to a large array of different proteins with diverse functions. I've seen structure-based phylogenies applied to viruses, for example, using the delicious-sounding double jelly roll fold of the major capsid protein to develop a model of viral evolution, and gives a link for that. As you can probably tell, it's still in its early stages and there are some limitations, but structure-based phylogenies can be a very powerful tool in answering some tricky evolutionary questions. I'm excited to see where this sort of thing goes. All the best, Daniel. P.S. Thanks for all you do at TWiV. I study virus proteins myself, but my background is more in structural biology, so this podcast has been super helpful in expanding my knowledge of viruses while I study. I've also discovered a love for viruses, so I hope to keep studying viral proteins for a long time.
After that episode, I was thinking back to that discussion, and I realized if you step back from virology and go back, several decades, structural phylogeny was also just called phylogeny.
Yeah, it's true.
Oh, good point.
Right? I mean, it's counting the hairs on the bug's butt,
Yeah.
and that's how it's this species versus that. But
Yeah, you're right.
applying
We've
that to
come
viruses
full
is
circle.
definitely new.
Yeah, I have three things to say. One, I stand corrected. Two, this is really cool. And three, Daniel, you write well.
I was about to say
Okay.
that too. What a wonderful email.
That's very
yeah.
I
good.
was going to say that as well. Yeah.
Yeah, so hear me out. You write well, and that is an extraordinarily useful talent, so don't neglect it.
Mm-hmm.
You can actually make a living on that.
I wonder who he could ask about that.
And of course, once you encounter viruses, you will love them. And encounter, I mean, when you start to study them,
Right.
not to get infected by them, because that's where the problem arises or can arise. All right, it's time for some picks, Dixon. What do you have for us?
Well, Vincent and everybody else, I'm back on my musical kick, and I forgot to mention this guy, and he's a remarkable guitarist. His name is Baden Powell, and as I wrote, he was named after the Boy Scout, the founder of the Boy Scouts, who happened to be, I think, English. Is that right, Alan? I think the Boy Scouts were.
You asked me?
No, well, I thought you're a fount of
Scout's
information.
honor. I don't really know.
Yeah.
I think he might have been English.
I think Baden Powell was. At any rate, you should watch this video. He's playing the theme from the Black Orpheus, and unfortunately, he's got a cigarette stuck out of his hand, and he's playing, and the smoke is going all over the place, right? And I looked up what he died from. He died from pneumonia. I'm happy that he didn't die from smoke-related illnesses, but at any rate, you should listen to this guy. He's just absolutely phenomenal, and he was Brazil's greatest musician, I think, bar none, including Antonio Carlos Jobim.
That's some stiff competition.
Well, he was widely respected as the progenitor of all of the popular Brazilian music that we now take for granted. So that's an uplifting kind of a musical
If he died of pneumonia, Dixon, that could very well have been
could have been
smoke
from
associated. When you said that,
It
I was
could
like,
have, that's no, you're quite right. You're quite
definitely
right. You're
could have.
quite right. You are quite right.
And yes, it was Great Britain, the Boy Scouts. I checked this. I
Yeah, I
checked
thought so, too.
1908, Great Britain.
And I have to tell you that my father used to own a Cocker Spaniel whose name was Zachariah Baden-Powell Condit.
Oh my God.
And was he a good guitar player also?
I just don't, I have no idea where his middle names came from, what my
Wow.
father was thinking at the time. He
Dogs
was not a
with middle
scout.
names are hilarious. People that give middle names to their dogs or last names. Like,
Angela, what do you have for us?
so today, I don't have anything specific. There's no website or anything yet. But as we actually a few weeks ago on the last TWiV that I was on, Rich, Vincent, and I went on this little rabbit hole about exercise. And we actually are planning now this podcast that we all said that we were going to do that wasn't really a thing yet. But when I was on the live stream 2 weeks ago, we discussed it. We also talked about like exercise and fitness, and lots of people had questions regarding this. So myself and Darren Elkreef, which is actually he's a PhD in exercise physiology. He did his PhD at McGill. He graduated last year. We are going to be, we are in the process of creating a new podcast on the platform of Microbe TV, and we have a name. I won't say it yet, but we want to hear from all of the listeners. Please, if you guys can send us things that you're interested in regarding exercise or regarding exercise science, something specific that you want to learn more about, or nutrition or sleep, or all of these different health and fitness in the realm of health and fitness, please send it. For now, we will have another email for myself, but as of right now, it's Vincent@microbe.tv, and he will forward me those emails or any of the information. So please send your ideas or anything. We'd love to hear what you guys want to hear about, and then the first few episodes, we can build around what the Microbe TV listeners would like to hear about. So we're excited about that. Yeah.
You are here to pump them up. Yeah.
Literally,
Exactly.
to muscle pump them up. Yes.
Cool. All right. Looking forward to the title.
Yeah,
It's
the
very
title's
cool.
really fun.
I like it very much. All right. Rich, what do you have for us?
I have some eye candy, a photo site called Mark Smith Photography. I've stumbled over some of his images and videos and social media, and so I finally decided to go to the source and see what there is. And this is an incredibly rich source of nature photography. And so, you can go, I gave you the top level directory, but go to gallery and go to videos and just spend some time there. The videos in particular, it says some just incredible slow motion photography of like ospreys picking fish out of ponds and stuff like that, or bald eagles doing the same thing. Amazing stuff. I will
love
say no
this
more.
stuff.
It's
I looked through some
just
of them
amazing.
earlier. So beautiful. I love these types of things. I could sit here for 3 hours looking at videos.
And he narrates. I just looked at one of the videos. I've never seen any of the stuff done like this. I just seen clips on social media. He narrates his own videos, and he's got one like 30-minute video about ospreys, and the guy is just nuts about ospreys, and he knows more than just taking pictures. He knows about how these birds think.
Awesome.
And
That's
how
so
they
cool.
operate. It's very cool, and he does a great job narrating them. It's just
Videos
wonderful
of
stuff.
birds of prey are so beautiful. I find them just like watching them in slow motion, especially.
I agree.
Yeah,
I totally
so
agree.
beautiful.
Cool. Alan, what do you have for us?
I have also eye candy with a science bent. So my pick is 2 links. There's an article in Science about it, and there's the actual site. So this just launched at the Florida Museum of Natural History. It's called Open Vertebrate, and it is a database. You can, if you're a researcher, you can go in and search the database for all sorts of stuff. If you're just like me and you want to look at the cool pictures, you can click on the specimen gallery get a sampling. They scanned animals, and these are 3-dimensional scans of all kinds of stuff. And if you go to they've got lizards, they've got fish, they've got all these vertebrates, and you've got 3D like medical imagery in color.
Wow.
It's just
so
really,
cool.
really cool to
This
look
is
at.
Another rabbit hole that I could spend hours on this website.
The science article has more of the images and describes what the project is all about, and this huge effort to get these specimens and get them scanned. And there are the challenges of how do you fit a Galapagos tortoise into an MRI, and they used an inner tube. And put it on its back, so it's there on the inner tube, just like lying there and being fed into the very cool stuff.
I feel a certain.
Did you know this?
I feel a certain amount of completely unjustified pride here.
Yes,
I'm intimately familiar with the Florida Museum of Natural History. It's not a big operation, okay, but it's extremely well done. And we used to go there fairly frequently. And in fact, years ago there was another pick from this museum, because they have what at the time was the second largest collection of butterflies in the world,
Really?
plus a live butterfly exhibit, the butterfly rainforest. That is,
wow.
I'll tell you, it's a destination. So good on them. Good for you.
I would like them to have the world's largest collection of Burmese pythons, but I don't think that's going to happen.
well, yeah, they're trying to do that.
You see, that's how you fit an MRI into an animal. The animal actually swallows the MRI, and then you take the picture.
This is so cool, though, Alan. Thanks for sharing. Yeah.
Sure.
All right, my pick is for Angela and any veterinary nerds who might be listening. This is veterinary apparel for veterinary nerds.
So
And
cute.
they have hoodies, sweatshirts, T-shirts, jackets, accessories. They have some cool stuff here. I saw a what is it? Toxocara canis
Vince,
T-shirt.
that's free.
All the
I'm sorry,
parasite
Dick.
things. And then one says, enjoy chewing your fingernails. And of course, like when you're in veterinary medicine, I mean, and all the time you shouldn't chew under your fingernails, but you're always told that obviously like little tiny parasitic eggs are under your fingernails. So you shouldn't chew, and obviously bacteria as well. But, and on the homepage, the little guinea pig with the glasses is so funny.
Yes.
That's so cute. It's so cute.
Ah.
Thanks for this, Vincent. I love it. And the
You're
little
welcome.
frog, all the ones with the little frog is so adorable.
They got mugs, they got water bottles, totes.
And the
Cool.
frog
Duffel
says,
bags.
tired of being a paratenic host? That's so funny.
That's very funny. Yeah, that's very funny. We also have
Cute.
a listener pick from Anne, an interview with Reed Jobs, son of Steve Jobs, who started a venture capital firm to support cancer research. Reed was studying to be an oncologist during his father's illness. He talks about why he didn't complete these studies, but also why he has returned to fund innovative research. From the description, quote, in high school, Reed Jobs was a summer intern in oncology labs while his dad, the late Apple's co-founder and tech icon Steve Jobs, was battling pancreatic cancer. In his biography, Steve is quoted as calling in his son's interest in biotech the silver lining of his illness. And making cancer non-lethal has become Reed's life mission. In 2023, he spun off the venture capital firm Yosemite from Emerson Collective, the philanthropy and family office founded by his mother, Laureen Powell Jobs, to focus on cancer research and biotech. Kara and Reed talk about the research to startup pipeline, how he's been influenced by both of his parents, and whether AI, mRNA, or CRISPR will be game changers for cancer patients.
Excellent.
That's cool. I didn't know that about
Cool.
Reed Jobs. And that is TWiV 1095, or 1095. Take your pick at how to say it. You can find the show notes at
1095
Micro.
sounds better.
You like
Sorry.
1095?
I like 1095.
Limited
For you,
time offer,
1100.
only $10.95.
For you, 1100.
Yeah, the show notes are at twiv@microbe.tv. If you have questions or comments, twiv@microbe.tv, or pick, you can send your picks in. We'd love to get them. And if you enjoy our work, We'd love to have your financial support. We are a nonprofit corporation, so you're in the U.S. Your donations are federal tax deductible. Go to microbe.tv/contribute. Dixon Despommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Oh, you're so welcome, and I love it. I just love it. New stuff every time.
All right. Angela Mingarelli's at McGill University and Immune Vet on the X, I suppose that is, or is it Blue Sky
Zitter.
or both?
Can we just call
On
it
X.
Zitter?
Zitter.
I haven't heard
On
of
X?
that. On X, yeah.
Thank you. Thank you, Angela.
Yeah, today was great. Thank you so much. See you guys soon.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Thank you. Always a good time. I love this stuff.
I agree.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.